In January 2021, DPHL identified the first alpha variant in the state. Beginning of March 2021, DPHL identified the first beta variant in the state.
End of March 2021, DPHL identified the first P1 gamma variant in the state. In May of 2021, DPHL identified the first B16172 Delta variant in the state. Currently, this variant makes up approximately 50% of our variants found from our sequencing. Genetic material is extracted from samples using an automated extraction platform, the Kingfisher. The genetic material from each sample is then broken into little pieces and then tagged with a unique identifier in a step called library preparation. It's like having a field of grass. Each blade of grass is given a unique identifier, and when examined together, all the blades of grass make up one field or one sample.
Then the samples go on the sequencer. The sequencers identify the pieces of each sample by the unique barcode and record each part of the genetic material of the little pieces of each sample. Using our previous example, the sequencer reads what makes up each blade of grass in the entire field that is the sample. Each sequencing run has multiple samples or multiple fields of grass. The sequencer completes the sequencing and outputs the raw data as a computer file. Our bioinformaticist then takes those files and reassembles the little pieces of sample back into a full genome. The assembled genome of each sample can then be analyzed for type of variant and amino acid mutations and deletions. Basically, we can now look at each field of grass as a whole and see what type of field it is. Variants are typically classified by their pangolin lineage. Pangolin is an analysis platform that is globally used to analyze and classify COVID-19 variants. Additionally, the WHO has recently assigned identification labels using the Greek alphabet to the lineages. All COVID-19 strains are variants. The CDC just determines which of these variants are of interest or concern. As per CDC guidance, variants of interest include B1427 and B1429 or epsilon, B1525 eta, B1526 iota, B16171 or kappa, and B16173. Variants of concern include B117 or alpha, B1351 beta, B16172 or delta, and P1, which is gamma.
Variants of interest and concern can exhibit increased transmissibility, increased disease severity, reduction in neutralization by antibodies, reduced effectiveness of treatment or vaccines, and/or diagnostic detection failures. Therefore, epidemiologically, it's important to track the variants of interest or concern circulating our state to guide public health response to the pandemic. It takes approximately 4 days from start to finish when the sample walks through the door to reporting results to the epidemiologist.
We can only sequence what we get sent to the lab, and we have been working with commercial labs to send positive specimens to DPHL. Also, LabCorp is sequencing COVID as well, so we are not the only ones in the game.